Now, we mustn't forget the biome. The biome means the flora within the whole of the alimentary tract from the mouth downwards. So improve the biome and increase the resistance to neurodegenerative diseases. So you think, well, why is there an issue between the gut and the brain? And the simple reason is if you've got a leaky gut, you've got a leaky brain. So toxins from the gut will cross over from the gut into the blood, will readily get from the cerebral circulation into the neuronal area and cause problems. There's nothing like a dose of upset gut. You know, for one reason or another, you've eaten something or you've got a bug and you feel awful in your head. You get a headache and all the other. It's the toxins coming through, which they do. So the microbiome disturbance is due to the food we eat. So it could be a contaminant, it could be because it's sterile, it could be because we swallow a lot of it down with chlorinated water. But the biggest source of the sterile gut and allowing then bad anaerobic bacteria and funguses is food preservers. Food preservers are a much greater problem to the humans than anything else, because food preservers are there to preserve the food. Okay, all those benzoates are there to preserve the food, but they preserve the food inside you as well. So in other words, if they're sterile, your meat can contain, be left in the fridge for weeks and weeks and weeks, your processed meat. Sometimes I buy a ready-made loaf of bread, not often, but sometimes if the kids are coming up, and the kids come back in a couple of weeks and the loaf is still fresh in there, and you think, if I have a loaf from Jill, it lasts about 2 days, 3 days, it's starting to get a mould on it, But how come a loaf of bread here, in a packet there, can remain fresh for weeks and weeks and weeks, even though it hasn't been sealed again? It's because it's full of preserver. And most of those preservers, when you eat the food, of course, are going to go into you, and they will preserve your gut. So they act as like an antibiotic inside your gut. So it's not just the food or the quality of the food, it's also the fact that they may sterilize the gut. Gut organisms contain 99% of the genetic material in our bodies. There's so many bacteria. The bacteria are so small in comparison to our cells, but there's more bacteria in the body than there are, more bacteria in the gut than there are cells in the body. So 99% of the genetic material in our body is actually in the gut, in the flora in the gut. It is a store of DNA. So it's actually a source when we need to restore some of our nucleotides. It influences the brain. Antibiotics and over-hygiene affect the biome and increase the risk of dementia. People with the greatest diversity of biome have the lowest incidence of Alzheimer's, which in a lot of third world countries, they know that people eat awful stuff and eat with the fingers, don't wash their hands and so on, but they don't get Alzheimer's. And directly they start getting hygiene coming in and washing everything and using preservatives in their food, then they start getting Alzheimer's. So diversity increases gut integrity. Gram-negative bacteria, particularly Clostridium, known as C. difficile, increases the gram-negative lipopolysaccharide, which creates inflammation and permeability. So it's the gram-negative bacteria which are the real issues. And when we come to look at free radicals, we'll see it's the gram-negatives which are killed largely by the nitrate reactive oxygen species, the nitric oxide and the peroxynitrites. And these inflammation and permeability are especially in autism, Parkinson's and Alzheimer's. Propionic acid, or a saturated carbon-3, is a metabolic poison. Now, all odd-numbered saturated fatty acids are poisonous to the body, but they can get incorporated into cell structures. And C3 is normally broken down to C2 or to C1 and burnt out. Now, C3 propionic, if it builds up, it actually inhibits the conversion of or the use of B12 in the mitochondria. So it's one of the indications of a high, or rather of a deficiency of adenosylcobalamin. It increases the omega-6 to 3 ratios, it alters neurotransmitters, it increases glutamate influx into the mitochondria. This is something in the old fatty acid kits. If you want a source of this, then there's no problem, just ask Julie when you make an order. Propionic acid, it's a short-chain fatty acid that lives down in the colon, and they should have even-numbered fatty acids there. So we're going to look a little bit later on volatile organic compounds, and a lot of these are the short-chain fatty acids, like acetates and butyric acids and so on. So therefore we need to get our biome back better, and at the moment we do a number of probiotics, the Bifidobacteria bifidus, which is an anaerobic one, which lives mainly in the colon. And then the lactobacillus are the aerobic ones that live mainly in the small intestine, and the Streptococcus thermophilus. So the thing, and we do a mixture of all those called the smart one. The thing that's gaining interest now is the fecal transplant. Now, I don't know whether you've come across this at all, probably not. But the fecal transplant is when you actually take the feces from a healthy person, so-called healthy person, a healthy bowel anyway, and you transplant that into a person with a bowel problem. And it produces absolutely miraculous effects, from diabetes to heart disease, all sorts of things apparently clear up. And somebody on the Bristol seminar was telling us how they actually do it with their own patients. She actually supplies this from her own feces. She encapsulates it and puts it in the freezer and then gives it to the patient. We won't go into any more detail about it, but apparently it produces amazing results. So, anyway, so the point of what we're trying to say is I think the future with probiotics is going to be looking at this, and we'll probably put a seminar on in the autumn, I think, because it's such an interesting subject, with human probiotics. Human probiotics are really the future. Now, these are probiotics we get. There's hundreds of different probiotics that go in, but getting a human fecal probiotic is going to be the future. I think you get much better results with getting the normal feces back into order if you use a human feces culture. I'm not suggesting you actually do the fecal transplant because we're yet to find somebody who's got a good bowel. And how you judge that, I'm not sure. So, okay, so I find this is a very useful point of therapy localizing for probiotics, because probiotics really are not part of the body. They're inside a tube, which is not inside the body. It's inside the body, but it's not systemic, if you like, is it? You understand what I mean? So the body doesn't actually, the brain doesn't monitor, has no idea. It can't tell you what's going on in the gut. So what you have to do is to create a little bit of a problem. So has anybody got a bit of a tummy problem? Anybody get a bit of wind and things? You can't have dementia and wind. So well, you can actually. It might be why you put your hand out. Okay, let's try over here. So do you want to bring the smart probiotic? So what I'm going to do is I very rarely find probiotics show up. Pop with your head out there. So again, if you want. Yeah. Usually the people will tell you they get indigestion, wind, irritable bowel, call it whatever you want. So you would have done your workup with the patient. You wouldn't just do this with the person coming in and say, I've got a bowel problem, hop onto the bench and then do the test. But you do the preliminary work to make sure everything's working all right. So pull your knee to your shoulder. Okay, that's good. Now what I do is I get them to therapy localize just with 2 or 3 fingers there, just about 3 or 4 centimeters below the umbilicus. Don't press. Okay, just touch. Just to make sure there's nothing in there which therapy localizes. Then you press in. Okay, and that really squeezes the small intestine, and that's when they go weak. Okay, now, if you now bring on the smart probiotic, this is a combination of all the ones that is just listed there, and pop that on there, keep pressing in, and you'll see what we've got is a nice strengthening to the composite. So when you strengthen the composite, then we give them singly to see which one or ones it is. So sometimes the composite is better than the single ones. So if we start with the single ones, acidophilus, that's the most common one of the lactobacillus. So you press in each time. So he strengthens nicely to acidophilus. Bifidus, remember, is the anaerobic one that lives in the large intestine. Push in. So that doesn't do anything. Bulgaricus comes from Bulgaria. It's one of the original yogurt ones, that from Bulgaria. KCI comes from the Caucasus Mountains, otherwise known as Kaffir. The interest in this is because people in those regions live the longest in Europe, or did. I don't know now. What was that? Plantarum? Plantarum. Very important with the immune system in the intestine. Rhamnosus. I don't know where that comes from. Push in. Streptococcus thermophilus. Push in. That's it, I think, yes. We do find, interestingly, some countries seem to predominate with some probiotics. America, we found, is salivarius. And I couldn't understand this at all, because there's certain food preservers which are permitted in America but not over here, okay, which sterilizes their gut beautifully, okay, but they're not permitted over here. Acidophilus certainly is the most common one, which is what showed with you. And I would say probably followed by bifidus pretty closely. It's usually one or the other. Sometimes people need the smart one, which is a combination of the two, particularly if they've had antibiotics. But the majority of people, it's that pothole again. If in your case you only need acidophilus, we give him acidophilus. Right? If you give him all of them, you'll basically just raise up the ones that he doesn't need, and the differential with the one that you do need will remain, the pothole will still be there. So it's like having a pothole in the road, tarmacking it, over the surface the pothole's still there, you just tarmac the pothole, you don't fill it up. So if you're deficient in acidophilus, then you go for acidophilus. Okay? So just use that one. When do you take it? The answer is last thing at night. Why? Because that's when your hydrochloric acid is at its lowest. Because you don't want a time when hydrochloric acid is at its highest, because it will destroy the bacteria. So it's actually high now because you're getting ready for lunch. Okay, so the closer you get to lunch, the higher your acid is getting, the worse and more stupid it would be to take the probiotic. We always suggest putting it into plain water, which means basically clear water from filtered or from bottled water, but no chlorine in it, okay? And to putting the spoonful, which is usually enough, a level teaspoon, into the water, stir it, and just leave it for a few minutes for it to become viable in order to activate it, rather like you would with yeast, and then swallow it down last thing at night, right? If you forget, you could take it first thing in the morning, again your hydrochloric acid, provided it's before 7, okay, because that's when hydrochloric acid starts kicking in with stomach time. Okay, thank you. So probiotics, good lot coming out now about probiotics and a lot of health issues, so I think we'll put a seminar on in the autumn time related to probiotics and the gut flora, and particularly with the human fecal ones coming in. So what I want you to do is to try, when you've got somebody with a gut problem, is to try and therapy localize here if they don't show up to probiotics off your general systemic points. So if you were to come in off a meridian point, or possibly however you get in, even the therapy localization to a pain, off a coloured acetate, etc., you may find the probiotic doesn't show. Very seldom does it show by looking at it from a systemic point of view, because the brain doesn't know what's going on in the gut, because it's a tube inside the body. You've got to go to the tube, and you've got to distort it by pressing on there, and most people with a dysbiosis get wind or fermentation. So what you're doing by pressing there is you're pressing the wind. So you're pushing it into the tubes, and that's what makes the intestine, when it distends, makes the muscle go weak. So that's why it's a more reliable test than just just doing it more systemically. Okay? So try that, and I think you'll find that probiotics show up a lot more than what we'd imagine. I used to have a friend in Italy who I used to teach years ago, and he said, I only feel good when I've had a good bowel movement. And I sort of laughed a bit about it at the time. This is 25 years or so ago. But I think he's right. When your bowels are good, you feel good, don't you? Because you feel cleaned out there, and you're getting rid of so much junk and toxins in there. When we find, often we therapy localize down where the problems are, it's always in the lower descending colon and the rectal area where you get these different fecal build-ups of funguses and molds, etc., down there, and parasites. Okay, so we've got just time before lunch to do the suprachiasmatic nucleus, or the SCN. So the SCN is something we've done before, but this is the starting point, I find, really for all examinations with patients. And this stands for the suprachiasmatic nucleus, which is the body clock. Okay, it's the master body clock within the brain. So all rhythms, and we know that everything works in rhythms. Those who've trained in Chinese medicine know that everything goes in 2-hour cycles. So you have 2-hour cycle when your stomach is higher than the spleen and the heart, etc. But what governs this cycle is really interesting. and because there has to be something which switches the clock on to begin with. And the switch that switches the clock on is the birth process. So at birth we change from a world of darkness to a world of light. And we have in our tissues certain compounds which are light sensitive, not just the back of the eyes here, but in the skin. So we go from a world of darkness into a world of light in a matter of a few minutes, depending on the speed that the baby comes out. But that exposure to the light is what has a profound effect on the suprachiasmatic nucleus in the hypothalamus. This enables the suprachiasmatic nucleus to start to function. So we're off, but there's no biological rhythm or function in that sense before the birth. So the switch that starts off the body rhythms, the circadian rhythm as it's called, is the contrast of dark and light. So this is why we now live in a different world. We abuse that by putting on lights, artificial lights in the evening time, okay, when it's dark outside. So this means that certain parts of our body clock go off because we should be asleep. The pineal gland saying, I should be awake now, and you should be in bed, and the light should be off. So as a result, we get out of sync. So we have to get back into sync, which is called entrainment. So all our clocks have to be reset. So the moment you open your eyes and exposed to the light in the morning, all your body clocks reset. This is called entrainment. So everything is lined back up again, ready for the next day. So some body rhythms will be more than 24 hours, and some will be occasionally less. But they all reset to the 24 hours with the contrast of light and dark. So this nucleus is a tiny region located in the hypothalamus. It's situated directly above the optic chiasma, hence the name suprachiasmatic nucleus. It's responsible for controlling all the rhythms. The neuronal and hormonal activity it generates regulate many of the different 24-hour cycles. It contains around 20,000 neurons. It interacts with many other regions in the brain and contains several cell types and different peptides, including vasopressin or antidiuretic hormone, a vasoactive peptide, intestinal peptide, and other neurotransmitters. It's situated in the anterior part of the hypothalamus. It sends information to other hypothalamic nuclei and to the pineal gland. So we know the pineal gland is the one that switches on and off, and it switches on when it's inhibited from darkness coming in through the eyes. So when we inhibit it, it actually activates the pineal to produce melatonin. When we stimulate it through our vision of light, through, as we'll see, the noradrenaline receptors, we inhibit the pineal from working. But it also, as a result, controls body temperature and the production of hormones, especially cortisol and melatonin. Many aspects of mammalian physiology show these rhythms, including sleep, physical activity, alertness, hormonal levels, body temperature, immune functions, and digestive activity. So here we go, hormone levels. So we do show, as we'll see, when you use the suprachiasmatic nucleus or the definitive meridian, you do find, if you cross-check to the organs in the kits, particularly the organs that produce hormones, the pineal gland comes up a lot. And this is usually when people aren't producing enough melatonin and have sleep issues. So the point is, how do you regulate that sleep pattern and stimulate the pineal? So there has to be something which helps as a hormonal modulator. And I'll show you what that is, because it quite surprised me what it actually is. A very simple substance and what it contains in. So the retinohypothalamic tract, this is the tract that takes the information back from the eyes to the hypothalamus, is a photoneural input. It's rich in melanopsin. This is the substance, melanin, which is in the back of the eye. So there's 3 types of melanin, which we have in the back of the eye, and these correspond to the genetics, or inherited from your mum and dad. So these we know will absorb red, green, and blue. And we all have a defect in one of these, just like you have different colours with your hair and with your skin. So red people, people who weaken to the red acetate, basically are overpowered by the red end of the spectrum because they don't have enough melanopsin in the back of the eye of that type to absorb the red wavelength. And so the red wavelength goes through and overstimulates the optic nerve, which is why they go weak. Green people, they weaken to the green, and blue people weaken to the blue. So this is why the theory is that people weaken to colours when it's put through the eye. So this is a light. When you put an acetate on a person from the outside, it's a light from outside, the sun or the light in the room here, going through the eyes And these light waves are, or can be waves or particles, and they're called photons. So when we're exposed to photons or packages of light, these are stimulating us from the outside. But this afternoon I want to talk about biophotons, which are the packages of light that we create inside from the mitochondria, and what we can use to measure mitochondrial function. So this is a good time to have a lunch break now, and then we'll reconvene, show you how to make the, to diagnose the definitive meridian for using the suprachiasmatic nucleus. This is the meridian, and we'll talk about this afternoon, is probably what you started with. Okay, the definitive meridian means the ultimate one in balance, if you like, with you. So when you found out what this is, it's probably been there since your birth. So it starts the moment you're born and exposed to the light. Okay, and you'll always find associated with that meridian a weakness of the muscle, and this will be your most, and we know that all meridians have emotions associated with them, and in that muscle will be the trigger point. And when you find the trigger point, we'll go for it with the thumb. Okay, and when you find it, it's quite painful to stimulate it, but boy does it release, the emotional side of things.